Okay, so I'm going to put this on setup because that's a baseline non-stand control. I'm going to change the file name to unstand sample. And then I say turn this on and acquire. See, there's a lot of E cell there. So it looks like I want to increase the gate. Oops, it's not changing. Now it is increasing. I want to gate majority of the cells are there. So that's the non-stand cell. So remember, FSC is cell size, SSC is granularity. That's cell. For some reason now we see very few dust. That's a good thing. So this is FSC cell size. That's channel 1. So I have channel 2 and channel 3. So both of the channel, you see the signal. You still see some baseline signal. There's no fluorescence. Those are just baseline signals. So you still see some baseline signal like 50. This is probably like also like 30 or 40. For this channel, the baseline is less than 10. Is 3. So that's the two-dimensional part. This is called a histogram. That's the FSC. Most of the cells are very large, so it's good. SSC, most of cell is kind of pretty rough surface too. Green channel, red channel, long run channel. It looks like there's some cutoff, but it's good because at the baseline, since you see there's nothing exceeded 10, so we know the voltage seems to be appropriate. You don't want to the baseline signal very high, and then when you take the actual signal, it's going to over the range. So I'm going to pause this, actually record this baseline. Now when we're recording, we want to go to the acquire storage. I want to make sure we are recording the right one. So here I'm recording, say, for G1 equals R1 for 10,000 event. R1 is this. R1 is this square window here. That's called gate 1. That's for R1 is for gate. Acquisition will stop when 10,000 of R1 events are counted. Basically, I will count any of 10,000 dots inside of this R1 window, and then this measurement will be stopped and the file will be saved. You have to make sure you check 10,000 usually the minimum. And for each cell, that's not a problem. For mammalian cell, very often people will have a problem to actually even get 10,000 for the signal they want. So that's okay. Click OK. I'm going to now click acquire. You can actually see how many events. That's why we put a counter here. You can, if something wrong, you will see the counter will stop, or you see 0 there. Acquire. Okay, you see the counter. 700, 1400, 21. When will it stop? It stopped probably somewhere more than 10,000, because you see also there's some dot there. Look at dust. But it's not counting the dust, it's only counting everything inside of there. So you see, now it stopped at 11,000. So there are like 11, there are about 1,000 outside of that.
Right, okay.
Yeah. Okay, good. So that's stuff. And then we put this one on standby. Now you need to keep track which tube you are working on. So this is the tube, and this will be the next sample. So you want to take the next sample with this live cell. Yeah. Yeah, we vortex them, so they won't step together. And then, oops, pull this to there. To keep track of things, we put this on top. And then and we want to do this as quick as possible. Pull the base aside and pull this tube immediately back. Pull this aside. Pull this one back. Put this one here and switch. I'm not sure how long it takes, but doing it as fast as you can. Alright, and now always remember you want to change the file name. Otherwise everything will be stored in the same file. In the end you still don't have all the data you want. Okay, so this file will be number 1 as live cell. But we will see whether it's live or not, hopefully live. So let's see. Like, I'm not sure right now, so I'm going to click setup just to see whether the signal is right or not. Okay, acquire. This is a live cell, so I should see no signal. I should see no red cell, very few red cells. That seems to be good. See? Red should be in channel 2 or 3.
Okay.
I see something in channel 2, a little bit, but majority of them is still in the very low range. So it seems to be this one is a live cell. So I'm going to pause this. And uncheck this and just store this data. That looks like a good signal. If there are so many cells, it stops immediately. It actually goes to 25,000. Okay, now we are going to do the second one. That will be half-live, half-dead, I guess.
So are all live cells going to be blue? Is that why that?
No, the blue is different thing. The blue is actually shows just to highlight the peak. It's not doesn't mean it's blue.
Okay.
That's this one. Keep track of the sample. Number 2. Okay. Now I'm going to change the file name. I'm going to say live deck. And then set up again. Apply. Now let's see. Oh, I forgot to put this one wrong. Let me see. Well, it's not half, but you see a lot. Yeah, you see now that this time you see that both F2, F1, there's a lot of cell had very strong signals. Yeah, here, these 2 channels, FL2, FL3. If you can picture in 3 dimensions, that's how it looks like. That's FL3, that's FL2, that's the cell size. So in this circle, that would be the dead cell. Still, it's actually probably a good thing. Even though we left all the cells there, most of them still seem to be alive. But these can live for months in water. Actually, we didn't even leave them. We leave them immediate, so that's actually even longer. So, okay, so I'm going to save this file. Okay, and it saves it immediately because there are so many cells there. Once I'm not running, I put on standby. For this sample, it doesn't matter, but for many of the precious samples, maybe make a difference. So if you look at this peak, it's broader peak now, and hopefully when we switch to the dead cell, we see a stronger peak there. Okay, I'm trying to keep track of the sample. Try to vortex the sample before you elaborate. because you don't want to. If you do vortex, you may have two cells stuck together, and if both of them dead is okay. If one is dead, the other is alive, it'll give you an average signal. That's kind of confusing. So for this one again, I'll try to do it as quick as possible. Sorry. And always remember to change the file name. And this one will be that. I'm going to just go ahead to acquire and see what happens. Oh, very good. That's also because I'm running out of and this peak does seem to be a little higher than that. Okay. Going to